Greetings, dear learners. In the last class, we were learning analytical toxicology. Under that, we learned how to store properly the samples collected and the bedside color test for the various poisons. Under that, we learned importantly that it is preferable that every sample is prepared in duplicate. One part of the sample shall be kept at 4 degrees Celsius to analyze it as soon as possible, and the another part shall be kept at minus 80 degrees Celsius for the long storage. Then, while learning about the different color test, we learned that when performing color test, it is always important to analyze concurrently with the test sample the reagent blank that is known to contain not to any of the compounds of the interest, and then the secondly a known positive sample at an appropriate concentration. Then, as we can see here in the picture, Trindler test, like that we learned the many tests. Here we can understand that this is a blank sample, this is the test sample, and this is the sample that already contains the substance of interest like salicylic acid. Here in this case, that showing the violet discoloration, and the test sample is also showing the violet discoloration. So this is suggestive of presence of salicylic acid in the test sample. We also learned various other color tests and their importance. Today we shall move for the other parts of the analytical toxicology. For the purpose of chemical analysis, poisons are grouped according to the method used for the isolation of the substances from the matrices. These are the noxious gases, volatile poisons, that includes organic and inorganic volatile poisons, then non-volatile poisons, that includes again organic and inorganic groups, then plant poisons, and the miscellaneous groups of poisons. The first step to the analytical toxicology begins with the extraction of the drug or poison from the large amount of the biological components such as proteins, lipids, nucleic acid, and the membrane. Like the way when we collect the sample, we collect either the gastric lavage sample or the blood sample or the from the postmortem examination, various tissues and everything we send to the toxicology laboratories. There, the main challenge for the toxicologist is the extraction of the poison or the drug from this large bulk of the tissues that are sent along with the poisonous content. So, the first step before analyzing the poison is their pre-treatment and extraction from the unnecessary tissues. So, the small amount of drugs and poisons incorporated into the human bodies are hidden among the large amount of the biological components such as proteins, lipids, nucleic acid, and membranes. It is not easy to detect only the target compound from such a complicated matrices. Before instrumental analysis, extraction procedure is usually essential and very important. Extraction methods are used for removal of such proteins and lipids existing in large amounts in the biological matrices. Also for removal of impurity compounds interfering with the chromatographic separation. For the condensation of the target compound and for removal of the compounds causing trouble in the instrumental analysis. So for all these purposes, extraction is performed. There are numerous methods of extraction according to the target compounds. The selection of proper extraction methods depends on the various controlling factors like the nature of the poison, matrix or matrices, and also the quantity of sample available or forwarded for the analysis. The active constituent should be extracted from the sample in minimum steps to avoid loss during the processing. The extracted material also require proper stripping or purification to avoid interference of matrices as far as possible. The efficiency of extraction and stripping determines the lower limit of detection, precision and accuracy in the determination. So, the analyst plays a significant role in the selection of proper methodology on the basis of different parameters like case history, amount of the physical state of the matrices, analytical requirements and also the infrastructure facilities that is available. The present table lists the various methods of extraction, both classical methods and the modern method for the different types of poisoning. The classical methods are more popular in Indian forensic science laboratories dealing with large number of toxicological cases. In India, the poisons cover a broad spectrum, unlike in the foreign countries where they have the limited incidences or limited spectrum of the poisoning. Still, we are slowly adapting for modern methods of the extraction. So, you can see for the gaseous compounds, for the classical methods are micro-diffusion, absorption or desorption. The modern method for gaseous compound is sensor-based gas analyzer, gas chromatography. For volatile inorganic compound is the Gutierrez method, mass Berzelius method, micro-diffusion, digestion with the specific reagents under the specific condition of the pH, crystal test or color test that will decide what method has to be applied. Then the modern methods for the volatile inorganic are microwave oven techniques for the digestion, followed by the ion chromatography using ion exchange resins, spectroscopy. Then for volatile organics, the classical method is distillation, steam distillation, diffusion. Modern methods are gas chromatography, mass spectrophotometry. Then for non-volatile inorganic compounds, the classical methods are dry and wet incineration, group analysis, electrodialysis, digestion under the appropriate analytical conditions, titrametric, paper and thin layer chromatography. Modern technique includes the micro oven technique for the digestion, followed by the ion chromatography using ion exchange resins. Then inductively coupled plasma atomic emission spectroscopy, ICPES. Then inductively coupled plasma mass spectrophotometry. Then for non-volatile organic compounds, the classical methods are the solvent extraction, Estes auto method, digestion with ammonium sulfate, sodium tungstate or related methods. Then the modern method for the non-volatile organics are HPLC, paired ion extraction chromatography, HPTLC, supercritical fluid chromatography, solid phase extraction, micellar extraction, affinity chromatography, microwave assisted reaction system, accelerated solvent extractor, sweep distillation, universal trace residue extractor, and for the anionic class of poisons, the dialysis, chemical digestion, paper and thin clear chromatography are the classical methods, whereas ion chromatography by ion exchange resins are the modern methods. So, here we can see that the modern method has advanced with a new and developed instrumentation. These modern methods or instruments are also helping in the analysis of the poison. So, some of them we will be talking with the extraction method and some of them we will be dealing while talking about the analysis part. So, now we will understand some of the different techniques that are used for the extraction. One of the good technique is the solvent extraction. Here, the solvent extraction is a method where a system of two immiscible liquid is required for the separation of the material. The active constituent should be unevenly soluble in the system, thereby facilitating extraction of the constituent from one phase to other. The efficiency of extraction is determined by the distribution coefficient, that is D. D is equal to total weight of the solute in the organic phase and divided by total weight of the solute in the aqueous phase. If one of the two liquids contains a solute and the system is shaken and then allowed to settle, some of the solute will be transferred to the other liquid. The amount that gets transferred will depend on the relative affinity of the solute for each of the two solvents, that is the relative solubility. This is the basic mechanism that is used behind the solvent extraction. And this relative solubility or relative affinity is determined by the D value, that is distribution coefficient. Greater the value of the D, the greater is the efficiency of the extraction. So, you can see this is the funnel or the flask that is used for the solvent extraction. Here the two different liquids, one of the low density and one another of the high density. So, this is mixed along with the solvent. Suppose solvent is present in one of the liquid, then they are allowed to mix, and once it will have affinity for some particular liquid. So, we will separate both the liquid, so they will get attracted in the particular liquid in larger concentration than compared to the other liquid. So, we may able to separate the different solutes in different liquids. Solvent extraction is a common technique in the forensic toxicology related to the biological matrices. Solvent extraction method has now been upgraded and made automatic with accelerated solvent extraction. In case of solid non-biological matrices, continuous extraction by the Soxhlate may be employed, that is continuous extraction. Then another technique is the distillation. The process involves heating a sample of liquid to convert it into the vapor, which is then allowed to flow in another location where it is cooled and it is brought back into the liquid form. So, here some of the solute may be remained in the jar or some of the solute may go with the liquid. In this way, we may separate the different solutes. The distillation method is very commonly used in the chemistry laboratories. So, you all must be aware about this method. The vapor modification of basic distillation process are used for a specific purpose like distillation under the reduced pressure, steam distillation, fractional distillation, sweep core distillation. For the steam distillation, a steam is passed into the solution and the aqueous distillate is collected by the condensation. Toxicants from the acidic distillation process includes the ethanol, methanol, phenol, halogenated hydrocarbons, cyanides. On the other hand, toxicants from the basic distillation process includes the basic drugs like amphetamines, methadone and also aniline, pyridine and nicotine. Then the fractional distillation is the type of distillation which enables us to separate a mixture of volatile liquid differing in the boiling points, like separation of a mixture of kerosene oil with mineral turpentine oil in an oil water emulsion may be separated by the steam distillation as both have different boiling points. Then distillation under the vacuum. This is another type of distillation which provides separation of thermally liable volatile compounds at low temperature without decomposition. Then sweep core distillation. Here it is a special case of distillation that is based on the preferential volatilization of the organic compounds, especially pesticides are separated from wild lipids or the plant extracts using a stream of inert gases and subsequent isolation of volatiles on cola traps or the solid adsorbents. Universal trace residue extractor, Unitrax, and accelerated solvent extractor, that is the highly automatic extraction system for the rapid extraction of multiple samples, work on this principle of sweep co-distillation. Then another technique for the extraction is the micro-diffusion. Here the micro-diffusion is a convenient and popular operation that facilitates toxicants in the gaseous or volatile form in blood, urine or gastric aspirates to be detected or determined after isolation by the various techniques. For the micro-diffusion, this Conway micro-diffusion disc is used. So, way, micro-diffusion disc has two separate chambers. One chamber is a narrower chamber on which the substance to be separated is added, and that diffuses out in the surrounding chamber. So, in this way, this micro-diffusion technique is done. Then another separation technique is dialysis. It involves separation of a crystalloid from a colloid by filtering through a semi-permeable membrane. The separation process may be accelerated by applying electromotive force, that is electrodialysis. Then another technique is the sublimation. Here, this is similar to the distillation, except the sample is a solid to begin with and is converted directly into the vapor and then brought back into the solid. Sublimation is applicable to isolate toxicants in a solid matrices like naphthalene, anthracene. They are the compound that sublimates. That is, they convert from solid to the vapor and then again back to the solid only. Then another important method is the digestion or the chemical treatment. This is commonly used in different types of forensic science laboratories. Sometimes the toxicants are separated on the treatment with acid or alkali or digestion or water bath or muffle furnaces, so that the digestion of the tissue occurs with the help of acid, alkali or on the water bath. So volatile inorganic poisons or phosphine, arsine and hydrogen sulfide are isolated from their salt on treatment with the dilute acids. Then on the similar line, microwave digestion is used. Here the matrices are digested with acid or alkali in the microwave oven to facilitate isolation of inorganic poison in organic matrices under a specific analytical. The interaction of microwave with matrices results in production of heat with rise of temperature, for which digestion of the tissues or the biological matrices occurs. Then another technique is absorption. It is a slow process compared to adsorption at the surface, involving diffusion of one substance into the interior of the adsorbent material. Toxic gases and volatile in oil are entrapped and enriched by the process using a tube containing diverse absorbing materials specific for a particular toxicants. Then another good technique is the chromatography. Chromatography, we will be detailing the process of the chromatography in the next class. The chromatography involves the separation of substances based on their relative affinity for two phases, one stationary and the another mobile phase. Substances which have higher affinity for the mobile phase are moved or carried along with it and thus separated from those with the higher affinities for the stationary phases. Then another good technique is the chromatography. Chromatography involves the separation of substances based on their relative affinity for two phases, one stationary and the another mobile phase. Substances which have higher affinity for the mobile phase are moved or carried along with it and are thus separated from those with the higher affinities for the stationary phase. We will be talking details of the chromatography, including column chromatography, paper chromatography, thin layer chromatography, high performance thin layer chromatography HPTLC, ion chromatography and gas chromatography in details under the section of analysis, describing different instruments and techniques in the next class. These are the classical methods applied in India for the extraction of the toxic substance from the biological matrices. Then certain modern techniques that has evolved over time to fasten the method process of the extraction. The methods include various methods in chromatography as well as a special extraction methodology like the ion pair formation, solid phase extraction, solid phase micro extraction, micellar formation. Then the special type of digestion as a means of isolation of toxicants through the chemical processing like microwave digestion, digestion by the plasma source for the inorganic samples, are also emerging for their consideration in the routine toxicological analysis. Thus, there are the various methods of extraction under the head modern methods, which are either underutilized or unexploited in the Indian perspective due to lack of infrastructure facilities and analytical expertise. As it has become very difficult to cope with much inflow of the exhibits, modern methods are replacing traditional methods of the extraction for the SPD analysis. This is in the case of Indian scenario, as we are already loaded with a large quantity of samples. So we have to use the SPD, color test and the other extraction methods to filter it out. And in those cases, we are applying the test or analyzing on the large masses of the biological matrices. So the chances of giving the positive result or showing the presence are quite in doubt because of the large other biological matrices are associated with the toxic substance. A few methods like HPLC, HPTLC, GLC, GC headspace, microwave oven technique have become methods of choice. The other methods including hyphenated techniques like HPLC GC, SPC GC, GCMS are under processing for standardization in Indian perspective. We will be learning details of some of the modern extraction methodologies in the next class, along with the details of the modern analytical instruments and the analytical procedures. Thank you for today.